Hi, 8th grade. This is your first video lecture, and our topic is scientific method because that's what we're going to be studying this unit. We're going to be learning about all the different steps of scientific method and how to, I just got a text, how to use scientific method to solve problems, answer questions. And that's basically what scientific method is. They are steps or a process that scientists use to solve problems and answer questions. So that's what we're going to be focusing on in the video today. And I'm going to go through each of the steps, and I'm going to use the freaky frogs to explain it. So if you've read this text, you know what I'm talking about. All right, so for the very first part of scientific method is that scientists observe and ask questions. Now in this book they have the question first and then the observation second. Typically a scientist is going to observe something and then that's going to create a question in their mind, right? So it's also important to make observations after you have a question. It's always good to gather information, maybe research about the topic to see if there's any other experiments or data that's out there to help you understand better so that you can later on form a good hypothesis and design and conduct your own really good procedure. Okay, but the first part of scientific method is typically making observations and asking questions. So in this case, they were noticing, these students were noticing in a local local pond area, I'm sorry, in a local pond area, they were noticing a lot of these frogs had deformities. And the word deformities just means that there's something wrong with the form or the structure, deformed. So some of them were missing a leg or maybe had an extra leg, things like that. So that was the observation that they made. And then their question was, what could possibly be causing those deformities, and is it maybe something in the pond water? All right, so let's move on through. I just want to do the steps with you. Okay, so once you have a question, once you've made observations and you have a question that you want to solve, the next step in scientific method is to form a hypothesis. Now, a hypothesis, a lot of people think, is a prediction. And it is in a way. You're predicting what you think the answer to your question is, or what you think the solution to your problem is. But there's one key difference in prediction and hypothesis. A hypothesis is a prediction, a possible answer to a question or a possible solution to a problem that can be tested. That is key, can be tested. So you can make predictions all day long about what's going to happen or what's causing something to happen, but unless you can test it, it is just a prediction. It is not a hypothesis. So in science, we have to be able to test our theories. We have to be able to test our hypotheses. That's what makes it a good hypothesis. Okay, so when the students were looking at these deformed frogs, they came up with some possible answers to the question of what was causing the deformities. Okay, the first one, hypothesis one, right here, was the deformities were caused by chemical pollutants in the water. Pollutants is something that pollutes or makes water bad, right? So some of the students said, well, maybe it's some kind of chemical that's in the water that's causing the frogs to be deformed. The second hypothesis is that they were caused by attacks from a parasite or from other frogs. Maybe there were some really aggressive frogs and they were like cannibalizing each other or eating their legs or something like that, or that it was some kind of disease like a parasite, a bacteria of some kind, maybe a protist, some type of fungus that was causing it. All right, and then the third hypothesis was that those deformities were caused by an increase in exposure to ultraviolet light from the sun. So basically like a sunburn or like skin cancer, some type of cancer that was caused because of exposure to too much ultraviolet light, which is the light that the harmful light of the sun's rays. Okay, so those are the hypotheses that they came up with. And actually these are all good hypotheses because they can be tested. Okay, but we're not quite done with the hypothesis. Okay, hypotheses have to follow a certain format when they're written, and we call it an if-then format. So we're going to take prediction 1, hypothesis 1, and if you notice here, they have written it in if-then format. Okay, so if you remember, let me go back really quick. The first hypothesis was that the deformities were caused by one or more chemical pollutants in the water. Okay, so this is the if-then statement they came up with. If a substance in the pond water is causing the deformities, then the water from ponds that have deformed frogs will be different from the water from ponds in which no abnormal frogs have been found. So they're basically saying if there's some chemical in the water that's causing the deformities, then if they compare that pond water to water from other ponds, that it's going to be different. They're going to find some different chemical that's in it that's not in a pond where there are normal frogs. Okay. Another way to write it down here, if a substance in the pond water is causing the deformities, then some tadpoles will develop deformities when they're raised in pond water collected from ponds that have deformed frogs. So here what they're saying is if it is a chemical pollutant that's causing some substance in that pond water, if they take that pond water and use it to raise from eggs, the tadpoles, everybody knows that frogs start out as eggs and then they develop into tadpoles and then eventually they grow legs and they lose their tail and their gills and they are frogs. Okay, so they're saying that if there's some chemical in the water that's causing the deformities, that if they raise from eggs all the way to frogs or from tadpoles all the way to frogs, that those frogs will also be deformed. Okay, then hypothesis 2, right here, it says if a parasite is causing deformities, then this parasite will be found more often in frogs that have deformities. So what they're going to do here is they're going to test the deformed frogs for evidence of parasites. So they're going to look in their tissue, probably under a microscope, things like that. And then the third hypothesis, which had to do with ultraviolet light, if you remember, they say if an increase in exposure to UV light is causing the deformities, then some frog eggs exposed to ultraviolet light in a laboratory will develop into deformed frogs. So if it's not the water, if it's actually UV light exposure, then they can take frog eggs and grow them in a laboratory being exposed to UV light, and those eggs will develop into deformed frogs too. Okay, so if you notice, all of these hypotheses are testable. Okay, not only do they predict the answer to the question or solution to the problem, but they can be tested. You could actually design a procedure to test them. Okay, now that's the next thing. You're going to test your hypothesis, and in order to test your hypothesis, you have to design an experiment that has only certain variables. Okay, I'm going to talk about variables, and then I'm going to finish up this video and I'll do the rest of scientific method on another one. Okay, so in an experiment you have an independent variable, you have a dependent variable, and you have controlled variables. Now that's different than a control or a control group, and we'll talk about that later on too. Okay, in an experiment you can only have one independent variable, just one. Okay, so what the independent variable is the one factor that the scientist is changing to see what happens, to see if the hypothesis is correct. And if you look at the hypothesis, okay, in the if portion of each hypothesis, that's where you're going to find your independent variable. So in this case, hypothesis 2, if a parasite is causing the deformities. So what's the independent variable? parasites or not parasites. Okay? In this one, if a substance in the pond water is causing the deformities. So in this, the independent variable is a chemical substance or not a chemical substance. Okay? And then the one down here, hypothesis 3, if an increase in exposure to UV light. So the independent variable here is exposure to UV light. Okay? So those are your independent variables. Okay? Another variable that you will have that they don't really talk about in here right now is the dependent variable, or the DV, the dependent variable. Now the dependent variable can also be considered the results, or the data that you collect, or observations. And that variable, the dependent variable, is stated in the then portion of the statement. Okay? So in hypothesis 2 here, again, your independent variable is parasites or no parasites. If it has parasites, it's going to cause deformities. If it doesn't have parasites, it won't cause deformities. Okay? So the dependent variable is here. The result is here. So they're saying in this one, if a parasite is causing the deformities, then the deformed frogs are going to have parasites more often. They're going to be visible. So your result, or your observation, if this is true, is that you would find more parasites in deformed frogs. That's your dependent variable, having more parasites, okay, in the deformed ones. On this one, if an increase in exposure to ultraviolet light is causing the deformities, then some frog eggs exposed to ultraviolet light in a laboratory will develop into deformed frogs. So the dependent variable here is whether or not eggs that are raised in a laboratory exposed to UV light develop into deformed frogs. So you're looking for deformed frogs or not deformed frogs in that. So the easiest way is to remember that the dependent variable is the result, or the outcome, and the independent variable is what the scientist is changing. Now, all other variables, in other words, all other factors that are involved have to be controlled. Okay, so we call those, I'm sorry to be doing this on the screen. Let me go back, sorry. So the variables are, a factor is anything that can influence the experiment's outcome. Okay, so if you're testing the exposure to UV light hypothesis, then that's the only change that you want in any of your setups. So like if you have an aquarium with frog eggs here, and another aquarium with frog eggs, and another aquarium with frog eggs, you want everything to be the same except the amount of exposure to UV light. That's your independent variable. So any other factor that might influence the deformities in frogs or the growth in frogs has to be controlled. In other words, it has to be the same for all of your aquariums that you have set up. Okay, we'll talk a little bit more about that in the next video. In the next video, I will go into the actual experiment, the designing of the experiment, what the procedural steps that they used, what were the variables that they controlled, what was the independent variable, what was the dependent variable, and we'll talk about collecting data and how to analyze that data. All right, so I hope this was helpful, and I'll see you in class.